Welcome Genetic Teacher Channel. Today, we are going to explain how to use spectrophotometer in laboratory for DNA and RNA quantification. Firstly, let's define spectrophotometer. Spectrophotometer, study the interaction between electromagnetic radiation with matter, to measure the concentration of solutes in solution, by measuring the amount of light that is absorbed by the solution in a cuvette tube placed in the spectrophotometer device. Using 1 ml purified water or T buffer as blank, to adjust the device to 0, then, add 10 microliter of each DNA sample to 990 microliter purified water or T buffer in 1 ml cuvette tube. When using cuvette tubes, take care for some items. 1. The cuvettes are expensive and fragile. 2. Be certain to only use clean cuvettes. 3. Do not scratch cuvettes. 4. Do not allow samples to sit in a cuvette for a long period of time. 5. Do not touch the base or the sides of a cuvette. 6. Make sure the cuvette is properly aligned during working in the spectrophotometer. And finally, 7. Wash cuvettes immediately after finish. Now, let's know the frame of spectrophotometer device and how it works. Firstly, spectrophotometer device consists of 0 to 100% transmission knob or button, plus wavelength knob or button, plus filter lever, plus mode knob or button, plus sample chamber, and digital display meter, as showed in the figures inside the slide. The light source, in the spectrophotometer, emits all colors of light, while the monochromator selects one wavelength that is sent through the sample, whereas the detector detects the wavelength of light that has passed through the sample, as presented in the figures inside the slide and next slide. And during working in spectrophotometer, follow these steps. 1. Turn on spectrophotometer power. 2. Allow about 5 minutes when first turned on for warm-up. 3. Fill cuvette tube half full with purified water or T buffer for blank. 4. Use 0 control knob or button. 5. Place the cuvette tube in its holder and close cover. 6. Fill cuvette tube half full with studied sample. 7. Place the cuvette tube in its holder and close cover. And finally, 8. Note the absorbance reading value from the spectrophotometer meter. For 50 microgram per milliliter DNA, take 0.25 milliliters stock DNA, to 9.75 milliliters purified water, or TE, buffer. On the other hand, for 40 microgram per milliliter RNA, take 0.2 milliliters stock RNA, to 9.80 purified water, or TE, buffer. After that, note the optical density, OD, 260 to 280 ratio. The ratio must be 1.8 for DNA quality, and 2 for RNA quality, but, be attention please, low ratio, mean residual phenol, or other reagent associated with the extraction protocol, and, high ratio, mean RNA, DNA, contamination. For example, if we have DNA sample diluted to 50x, and the reading of the spectrophotometer of this sample, after 50x dilution, was 0.7 nanometer, at OD, 260, what is the original concentration of DNA sample? The solution of this example will be done by following the next equation. DNA concentration per microgram per milliliter, equal, 50 microgram per milliliter DNA, multiplication by, OD, 260, or, OD, 280, multiplication by, dilution factor, and divided to, 1000. And according to this equation, the final results will be, 1.75 microgram per milliliter DNA. In the same line, pure DNA concentration equal, OD, 260 minus, OD, 320, multiplication by dilution factor, multiplication by 50 microgram per milliliter DNA. While DNA yield equal, DNA concentration, multiplication by total sample volume. On the other hand, the DNA purity of OD ratio, for 260 to 280, equal, OD, 260 minus, OD, 320 divided to, OD, 280 minus, OD, 320. This gel showed mark of DNA smearing, or DNA degradation, and mark of RNA contamination. Finally, the applications of spectrophotometer are 1. DNA, RNA and protein quantification, 2. Chemical analysis, 3. Food safety analysis, 4. Blood analysis, 5. Residual pesticide analysis, and finally, 6. Residual chlorine analysis. Thanks for your attention. Please like and share our video and subscribe Genetic Teacher channel. For any question, please comment us.